All right, we'll get started now that Howard's here. Everyone, welcome. I am thrilled that we were able to pull this off and bring everyone together for our third annual New Frontiers seminar. Many of you have attended our seminar last year, which was about CHIP, the year before about brain tumors, and this year we are talking about multimodal, multi-omics, and AI smushed together. Where are we going? What are we doing? And how does BLOODPAC get involved? Just going to run through some housekeeping quickly. The way this session is going to run, it's going to go for 2 hours. I promise we will be done at 4 o'clock. Number 2, we are going to have Peter introduce the topic in a more detailed fashion than I just did. Then we're going to have 4 15-minute presentations by some phenomenal individuals. Then we are going to have a 30-minute panel discussion. Welcome the speakers and 4 additional individuals to come speak on the panel, give some background about what they're doing in the field and in the space. And then we're going to open it up to about a 20-minute Q&A at the end. If you have questions so they don't leave your mind and you forget them, please put them in the chat. We will be collecting them and curating them and making sure we answer as many of them as we can. Please don't take yourself off mute, as I mentioned earlier, unless you have to. Some of you towards the end of the Q&A, feel free to scream stuff out, I guess, if you get really, really excited. And I will be giving everyone about a 5-minute warning at the end of the session to make sure that we're all, time sensitive and willing to get everyone out of here on time. Does anyone have any questions before we begin? Really, that's to you, Peter. You good to go?
Absolutely.
Thank you.
Great. Thank you, Lauren. Actually, it is a real treat to just scroll through the Zoom names. I would say this is all my favorite people in the whole world. Super cool. So anyways, thank you everybody for joining. Excited for this session. Some of it seems really, really obvious, but the fact that many of us have been dreaming about this world, however long ago, but I think we're getting there. We are well on our way. I always love starting off with this slide, just to give a frame of reference. Liquid biopsy first discovered in 1868. It's taken us a while to get to where we are today, but I think the pace of progress at this point is striking. Of course, then ctDNA evidence generation has really excelled through the next-gen sequencing progress that has been made, with, again, a lot of people working in this space. Why do we need to go beyond just one parameter? And to some extent, it's obvious, but it's always fun to put out some numbers, right? It's kind of obvious that we need to take ctDNA together with CTCs. It's kind of obvious that we need to take some other parameters into consideration. But when we look briefly at this slide, I just find this totally fascinating, right? That a lifetime risk for a woman is 1 in 8. Most of us have this number in our head, pretty straightforward. When it comes to younger women, 40 to 49 is roughly 1 in 65. If we compare that over to pancreatic cancer, that's somewhere in the ballpark of lifetime risk of pancreatic cancer for a woman. Or 1 in 48,000 in that same age range. For those of us living more in the math world of things, right, whether I'm looking at 1 in 65 risk or 1 in 48,000 risk, that just changes the game, and we just need to take all of that into consideration. And I think that is the world that we are entering right now, all of us together, which I think is super fun. Then, of course, there's the pathology, the foundation model breakthroughs that are happening on a very, very rapid pace. The fact that they are now, I'm not just putting the names, the years down, but actually I have to put Q1 down. So Q1 this year, right, Virchow model, super cool. Microsoft together with Page AI, and striking, because what that means that an enormous amount of content exists within just that H&E slide. And again, for all the anatomic pathologists in the audience, yes, you have been telling us this all along, right? But it's super cool to be able to put some real models, some real numbers behind this. And again, it is just striking. Fast forward to what Kuhn and his colleagues now did, Q3, and that is that these models, they can perform at a level that is equivalent to when we actually add genomics and imaging into it. So it's absolutely striking. And of course, what we need to do is bring all of this together in a much larger total picture, right? So we have what we draw our cartoons on the left-hand side, and then of course we want to understand the whole patient on the right-hand side as that patient goes through this journey of screening for cancer in the personalized level, and then diagnosing and treating that patient. And of course, the only way to actually do that is for this audience in particular is of course with our favorite liquid biopsy. And now whether that is peripheral blood, portal vein, pulmonary vein, CSF, bone marrow aspirate, aqueous humor, CSF, any one of those. And the data that's coming out again is super fabulous. So now there's a lot of other analytes, of course, right? There are proteins, lots of other aspects that many of us would consider our favorites because they speak to function, etc. So that's why I'm particularly excited that we can kick this off today with a fantastic set of speakers that will, as Lauren was saying, four rapid fire presentations to go through. And again, each one of them very familiar, I think, to all of you. And we have a lot of data out on the web, so I won't be doing any big introductions on this. And then Jim Goodsey will actually take this into a panel discussion to talk through this context, and then we roll from there. So with that, I'm going to actually close that, and we are on time, which I think is super important because again we have an enormous amount of content and ground to cover. And I will hand it over to Sam Hanash to actually talk about how proteins factor into this. And again, I've seen a preview of Sam's talk. I absolutely love it. And so Sam, please take it away.
Great, thank you, Peter. Do you see my screen?
Yes, we see your screen in the regular slides.
Wonderful. Right? So my group has long-standing interest in using proteomics to identify circulating markers related to different cancer applications. My short talk today is around circulating proteins for risk assessment. And so by way of disclosures, I have a lot of IP filings for single cancers, multi-cancer biomarker panels, and I am the one of the inventors of CancerSeq. I contributed the protein part of CancerSeq. So what about using protein biomarkers for risk assessment? Why would we want to do that? Well, we want to identify who is at risk and what cancer are they risk for. And you would say that right now we do have screening modalities for cancer, for common cancers, lung, colon, breast, prostate, and cervix. And so do we really need markers for those cancers? In Asian countries, they have screening for gastric cancer, but we don't have in this country. And the reason is not prevalent in this country like it is in some countries in Asia. On the other hand, if we could enrich in the general population a subset that is just as much at risk as in Asian countries, then for that subset of population, then screening for gastric cancer would be justified. So is there a problem with our screening modalities at the present time for those 5 cancers? The answer is there's a lot of concern that people are developing cancer that are not eligible for screening. Here's the headline in the Washington Post: "Cancer among younger Americans is on the rise," new study shows. The New York Times: "More young people than ever will get colorectal cancer this year." So clearly, coming up with criteria for screening that rely on age seems kind of like a thing of the past to me. We have to come up with better criteria to decide who would benefit from screening and for which cancer. So the concept that we have behind our approach is that there are cancers for which screening is available, but we would like, based on biomarkers and subject characteristics, determine somebody's risk for those cancers, so we could tailor screening according to risk. And if you can determine somebody's risk and they have an imaging modality that is sort of not very conclusive, then your risk assessment could help determine what would be the next step in evaluating these indeterminate findings, like, for example, in lung cancer. So what about other deadly cancer for which screening is not available, like the example of gastric or pancreatic cancer? So if we can identify subjects at increased risk for those cancers, then we could implement screening or monitoring for that subset of the population found to be in risk. There's a very important issue also related to identifying subjects at risk. Namely, if you are at risk, then there's an opportunity for preventive intervention. Like in the next few years, I'm betting that we're going to have vaccines available, and there's going to be a lot of trials with vaccines for subjects at risk for this or that cancer. And so why proteins among all the different types of omics? Well, one gene does not encode for one protein. One gene encodes for a protein that could have a multitude of different types of modifications, up to a couple of hundred different modifications. And some of those modifications, like protein cleavages, shedding of receptors, like chemical modifications, like glycosylation, and numerous other protein modifications, may be restricted to cancer, and they will inform about cancer risk. And so where would the proteins in circulation be coming from that would inform about cancer risk? Maybe from tumors at a very early stage of tumor development, from the microenvironment, exosomes being released from the tissue, or simply the host response to the occurrence of early stages or precancer in relation to tumor development. And so how are we doing our proteomics work to discover risk markers? We have had access to well over 1 million subject cohorts that had blood collected over a period of time, and we have accessed blood before they were diagnosed and asking 1 year, 2 years, 5 years, what can we find in their proteome, in circulating proteome, that would be indicative of risk. The one particular cohort that we have worked with extensively and we are very much in love with is the NCI prostate, lung, colon, ovarian cohort that we have done a lot of discovery work and a lot of blinded validation work with, and I'll give some examples. In addition to this, at MD Anderson, we have launched a number of cohorts of our own: LEAP, the lung cancer screening cohort; MERIT, the breast cancer mammography screening cohort, that have enrolled well over 10,000 subjects. And the subjects that were screened, we have access to their subject characteristics, their imaging findings, raw imaging data, and obviously the outcome, in addition to blood that has been aliquoted under standard operating conditions. And so what's interesting about the lung and the breast cohorts is that there are as many non-lung, non-breast cancers diagnosed as there were lung and breast cancers. So it would be very relevant to multi-cancer risk assessment and early detection, obviously. And so how are we doing our discovery? Well, we have a farm of mass spectrometers that's generating a lot of mass spec data for all of those protein forms. Some of them would be restricted to cancer. And you could imagine, with petabytes of data that we have generated, we have to have a data pipeline and more reliant on AI to be able to find patterns in the data at very early stages before somebody has been diagnosed with this cancer or that cancer. And so I won't have time, obviously, to cover every cancer that we've worked on by detail. The flagship cancer that we have worked on is lung cancer. And this is work that's been going on for 15 years, if not longer, that has led to the identification of a 4-marker protein panel for lung cancer risk assessment on the one hand, and for management of indeterminate nodule found during screening on the other. And so why lung cancer? Obviously, this is the #1 cancer in terms of mortality. 67,000 men and 60,000 women were estimated to die of lung cancer in 2023, and representing 21% of all cancer deaths. So this is kind of the blockbuster that we have been working on. And so, but you say that there's screening available for lung cancer. Well, the screening is limited to only a subset of, say, smokers or subjects at risk for lung cancer. And even those who are eligible only a very small percentage, like 5 to 8%, are actually being screened. So there's 40+ million subjects, ever smokers, that are at risk for lung cancer, and hardly a very, very small percent of them going through screening. So if you could identify, based on a biomarker profile and the subject characteristics in terms of smoking history and what have you, who is at a particular risk, even if they are not eligible for screening, I think that would be very beneficial in terms of impacting mortality due to lung cancer. And so, like I said, we've been working on this for a long time, and the landmark study that we had was when we were also part of the Mouse Models Consortium. That was an NCI mouse model consortium back 10, 15 years ago. And we were in charge of biomarkers as part of that consortium, working on just about every mouse model there. And we asked early on during tumor development, what can we find in the blood? And so that led to the identification of an immature form of surfactant B that we call pro-surfactant B, that's turned on very early during tumor development, and is turning on because NKX2.1, which is the proto-oncogene early during lung cancer development, is turning on this particular form of surfactant B. What was tantalizing for us is in one of the blinded validation studies that we did with the pan-Canadian lung cancer screening cohort, was that there were 17 subjects that had a negative CT. Before they were due for the next CT, they were diagnosed with lung cancer. And actually, they were diagnosed with advanced stage lung cancer. And this is the second group on this slide here. And we had blood that was collected at the time the CT was negative, and we were blinded in the analysis, but we looked at the pro-surfactant B data, and it turned out that that group that was destined to be diagnosed with lung cancer, that had negative CT, had very high values, as you can see here, of pro-surfactant B. So that became sort of like an anchor marker for us, that we could look at any other type of marker and see what do they add to pro-surfactant B in terms of lung cancer risk assessment. And so there's been extensive validation of pro-surfactant B, not only in the pan-Canadian screening cohort, but many other cohorts that we have worked on. And so when I say this is a sort of anchor marker, what have we tested in addition to pro-surfactant B? We have looked at the whole laundry list of other things in the blood, and we came up with all kind of combination, microRNA with an autoantibody, with pro-surfactant B and ctDNA and what have you. But at the end, we sort of had to be realistic that you cannot come up with a hodgepodge type of combinations if you want this to be implementable in the clinic, and if you want it to be sort of with minimal requirements and minimal cost type of thing. So we decided to stick with proteins and see what we can do with proteins. And to make a long story short, we came up with a marker panel out of 50 markers that we have tested to see which ones add to pro-surfactant B. We came up with a combination of 4 markers, pro-surfactant B, CA, C125, and CFRA, that we did extensive validation on. One of the validations that came out in JAMA Oncology in 2018. Based on that validation study with a very, very large cohort, then we went to the NCI requesting samples from the PLCO cohort, blinded samples, and funding to do the work. And we were successful in obtaining 10,605 blinded plasma samples from cases as well as from controls, representing adenocarcinoma, squamous, and small cell, to do a validation of our 4-marker panel for risk assessment of lung cancer. To make a long story short, the performance of the 4-marker panel, when we compare to the performance of the current screening criteria, the performance of the 4-marker panel with subject characteristics was much superior to how we screen today for lung cancer based on 20-pack years, or how we screened before based on 30-pack years. And so we're able to look at 10+ smoking below the 20-pack years of smoking criteria to see if that particular combination of markers would be beneficial. And the upshot is that we would identify 9.2%, based on the PLCO data, 9.2% more subjects that would be benefiting from screening with low-dose CT, and we could reduce the referral by some 13%, making it effective and cost-effective for screening for lung cancer. And so obviously the gold standard that everybody wants for a marker set or a marker panel for screening is, does it have any impact on mortality? And we were fortunate using the prostate, lung, colon, ovarian, NCI cohort that with the long follow-up, we knew who died in that cohort and who died of lung cancer. And so we asked, had those subjects that died of lung cancer gotten that blood test with 4-marker panel, would it have had an impact on mortality? And the answer is absolutely yes. And what you see in red here is the cumulative incidence of lung cancer death in the PLCO cohort, and how our marker panel, had it been used, and we know that a low-dose CT would reduce mortality due to lung cancer. Had they gotten low-dose CT, their mortality would have been reduced quite substantially. Now, is this a random finding here? No, because when you look at mortality due to other cancers, there was no impact of the 4-marker panel that is specifically designed for lung cancer. And so you can look at a single blood test and determine somebody's risk. But what about if that person has had blood done on a regular basis, like every year going for a checkup, and you're a never smoker? Well, get this blood test. Then you are able to determine a trajectory that would may be beneficial. So we looked at the PLCO data because there were more than one sample collected on a regular basis from the PLCO subjects, and asked, well, would the trajectory be beneficial? And the answer is yes, in a statistically significant manner. What you see here is the performance of a single marker. This is the blue curve compared to having a trajectory. You can move raising a red flag by at least a year when you have a trajectory compared with a single marker panel at one time point. So if this marker panel is useful for assessing risk of lung cancer, what about somebody going through CT screening and finding indeterminate nodules? Would that be beneficial? And again, we did validation studies of the 4-marker panel in relation to assessment of indeterminate pulmonary nodules. And what you see here is the performance of the 4-marker panel compared to the current sort of criteria for deciding whether a nodule is malignant or not malignant. And what you see is a shift with a very, very high specificity, improving the sensitivity if you add the 4-marker panel to whatever you know about that nodule in that one patient. What about small size nodules that usually tend to get sort of dismissed, like a nodule less than 6 millimeters? Well, don't worry about it. Maybe get another CT a year later or whatever. We looked at the performance of the 4-marker panel to rule in even very small nodules as to whether they are likely to be cancer. And you can see the performance in red here. Very, very high sensitivity, very high specificity in saying, no, your nodule, even though it's small, it's very, very, very likely to be lung cancer. So this is all about lung cancer. What about other cancers? So we have a similar story to tell about pancreatic cancer that led to the identification of the 3-marker panel for risk assessment for pancreatic cancer. And what you can see here, the marker panel consists of CA19.9 that we all know about, but also we have added LRG1 and TIMP1. And you could see the shift if you have blood collected a year or 2 years prior to diagnosis. You can see the shift in the curve indicating, raising a red flag that that person is at high risk for pancreatic cancer. And if you can detect pancreatic cancer a year earlier than with clinical symptoms, the impact on mortality would be quite substantial. And so lung cancer, pancreatic cancer, how many cancers to test for risk assessment? And so what we have come up with is a multi-cancer screening test, MCAS, that is intended to determine risk for 9 common cancers so that you could personalize screening according to risk for those cancers. Obviously, for those cancers, we already have a screening modality. For the others, we don't have a screening modality being applicable to the general population. And those 9 cancers are lung, breast, ovarian, esophageal, gastric, colorectal, liver, pancreas, and prostate. And so when you are amassing basically a certain number of markers that each is more or less related to a particular cancer, would a cancer like lung cancer stand out in the middle of a large number of biomarkers? And the answer is yes. So the 4-marker panel among multiple markers that we have in this panel stands out, as you can see here in green, for lung cancer. So it's not like, okay, well, your marker panel is positive, but we're not sure what cancer it is. For a lot of those cancers, we're able to identify the organ of origin for that cancer for which that subject is at risk. So the take-home message here is that multi-cancer risk assessment allows tailoring of screening based on risk for common and lethal cancers, whether or not there's a screening modality available. And it allows not only catching the cancer early, but also intervention, because that person is at risk. They may not have the cancer, but it would allow you to intervene for cancer prevention. And if you cannot prevent it, then detect it early. And also, I have shown here very, very briefly, is that establishing a trajectories, like, for example, is being done today for PSA. If there's a change in values in the marker panel for those 9 cancers, that would also send a red flag that that particular subject is at risk for that cancer. So we feel that in the field of early detection, the first step is identifying subjects who are at risk, because that would allow you to detect the cancer early, and better yet, prevent the cancer with what is available today and what's going to be available, hopefully, in the near future. So it's a very long list of names here that you have for acknowledgement, so I won't go through all of that, but we are very basically fortunate for the support that we got from the National Cancer Institute, from DOD, from our CPRIT Texas support here, and a multitude of foundations over the years. And happy to answer any question at any point in time. Thank you.
Thank you, Sam. Much appreciated. Let's switch to Wendy right away, please. Wendy, if you don't mind taking over the sharing screen. Wendy Winkler, Chief Scientific Officer for Droplet Biosciences, and she will take a different tag of how complementary data elements will actually help us even in low burden environments. So Wendy, it's all yours.
Thank you. Liquid biopsies that measure circulating tumor DNA are now being employed across the course of a patient's cancer journey, being used for diagnosis and for monitoring. But as powerful as this technology is, it's still very challenging to detect ctDNA in very low tumor burden settings. There simply aren't enough shed molecules in a tube of peripheral blood. This includes 2 settings where having more accurate information about disease status could have meaningful impact on outcomes. So early detection, as Sam was just describing, as well as the identification of residual disease immediately after surgery. So we need strategies for how we can boost that signal in the lowest tumor burden settings. And one promising possibility is to incorporate complementary data types and employ sophisticated computational models. So this could include readily available data like clinical data from a patient's medical record or pathology results from tumor tissue. This could also be a more comprehensive liquid biopsy that incorporates other analytes from the same sample. For example, RNA or metabolites or proteins like Sam was just talking about. So these alternative biomarkers, which may be tumor intrinsic or tumor extrinsic markers that are reflecting of the tumor's altered microenvironment, may be more powerful to detect the cancer that remains because they're maybe more abundant than shed DNA in very low tumor burden settings. And then we can use powerful machine learning approaches that have been developed to integrate these diverse types of data sets to identify new patterns that are associated with cancer. So today I'm pleased to share with you 2 examples of pre-commercial assays that are being developed by diagnostic startups that are looking to improve test sensitivity in these types of low tumor burden settings. So the first example is from my company, Droplet Biosciences, where we're testing circulating tumor DNA in proximal surgical lymphatic fluid and using those results along with traditional tissue pathology to help clinical teams to better tailor precision adjuvant therapy decisions. So in head and neck cancer, patients with localized tumors are generally treated by the surgical. And when the tumor is removed, a drain is routinely placed at the surgical site to reduce fluid buildup there. And this fluid is a mixture of blood, lymphatic fluid, and interstitial fluid. But at the 24-hour time point when we're sampling, tends to be mostly lymphatic and interstitial. So staff on the surgical recovery floor standardly empty those drains every few hours. So instead of discarding the fluid, we instead provide an ambient stable transfer fluid, then use a patented process to extract cell-free DNA from that fluid. And then we take DNA from tumor, blood, and the lymph and sequence them using a 699 gene custom hybrid capture panel and analyze them in a tumor-informed fashion. We applied this test in 42 HPV negative head and neck cancer patients who had both matched surgical lymph and plasma collected 24 hours after surgery. So on the left, you can see that patients with detectable ctDNA in the proximal lymph are significantly more likely to have disease recurrence than patients who are undetectable. Then on the right, we find that peripheral plasma does not reflect the recurrent status at this very early post-surgical time point, and this is consistent with what's been published previously. In head and neck cancer, decisions about a patient's adjuvant therapy course are currently made about 3 weeks after surgery and are based on a set of high-risk pathology features. So as you can see in our initial cohort of 73 HPV negative patients here, patients who are positive for these pathology features, for example, having positive lymph nodes, tend to have worse outcomes, but the effect sizes here are fairly small. In large part, this is because pathology can only evaluate tissue that's removed from the body, so cannot really predict based on information that's left behind. And the result of this is that recurrence rates are still unacceptably high. With up to 50% of patients in head and neck recurring within just 3 years. And we know that escalating treatment, for example, from radiotherapy to radiotherapy plus chemotherapy, can improve overall survival, but it's not done for everyone because the side effects can be so serious. So we need a better way to be able to identify those patients who are most at risk to selectively intensify therapy. In this analysis, we take 5 of those high-risk pathology features that are part of guidelines, and we combine them using a simple machine learning model. And see, not unexpectedly, that patients who are positive in this model are more likely to recur than patients who are negative. However, we see that this pathology model alone underperforms compared to the molecular data we see from lymph ctDNA. Now, if we take that 4-feature pathology model and add lymph ctDNA as a new feature, we find that the combination of these features is superior to either pathology alone or molecular data alone. So given the potential synergy we're observing here between pathology and ctDNA, we wanted to test whether it was possible to use lymph ctDNA to further stratify recurrence risk in pathology subgroups. Specifically, today, under NCCN guidelines, patients who lack the highest risk features, such as extranodal extension or positive margins, but who do have at least one other adverse pathology feature, are recommended to get radiotherapy and then to consider systemic therapy, such as chemo. So when we look at the 44 patients in our cohort who fall into this intermediate risk group, we see that patients who are positive for lymph ctDNA at 24 hours are significantly more likely to have disease recurrence than those patients who are undetectable. Additionally, when we look at patients who received radiotherapy alone, but later went on to have a recurrence, 100% of those patients were positive for lymph ctDNA at 24 hours. So this suggests that there's potential to use a molecular test to further stratify patients who are characterized by pathology and identify those that might benefit from treatment intensification. So in summary, we show that postoperative lymph is a novel proximal analyte to look for residual disease in the immediate post-surgical window when adjuvant therapy decisions need to be made. Additionally, this lymph ctDNA is significantly associated with recurrence, outperforming both time-matched plasma and pathology. And a machine learning model of ctDNA plus high-risk pathology features suggests there may be synergy between molecular and pathology data that could be used to improve risk prediction. The overall accurate MRD identification in the early postoperative time point has potential to augment traditional pathology to better personalize adjuvant treatment decisions. So the second example I'd like to talk about today is the work of this excellent team here at Prognomic. Just want to be very clear, I was not involved in the study, but I'm a member of their scientific advisory board, and I'm excited about the potential of their approach to early detection by using multi-omics and a machine learning-based classifier. So Prognomic studied over 2,500 patients with and without lung cancer who were enrolled across 77 clinical sites. They collected 3 different blood sample types per subject, and each of those was optimized for specific analytes, including proteomics, RNA-seq, metabolomics, and targeted immunoassays. This study represents one of the largest untargeted multi-omic discovery datasets in cancer to date, with more than 800,000 total features that were profiled across the full cohort. Such a vast data set is a fantastic source to perform AI-based feature selection, which they did to train a classifier model, and then they tested that classifier in a held-out independent validation cohort. Here is the performance of that classifier in the training set. The pink curve is the classifier trained on only protein features, and it achieves an AUC of 0.91 for all-stage lung cancer versus non-cancer. To investigate if multi-omics data could further improve lung cancer classification, they also trained a multi-omics classifier, now in black, using analyte features from all 4 of the analyte classes that they looked at. And this final multi-omics classifier had an all-stage lung cancer versus non-cancer AUC of 0.96, suggesting that adding additional types of analytes beyond protein improved classification. They next assessed the sensitivity and specificity of this classifier on their held-out validation set, which was almost 400 patients. They find that both sensitivity and specificity were 89% in this overall validation cohort. Notably, sensitivity was high across all the lung cancer stages, including the historically difficult stage 1 cancer, where they were able to see 80% sensitivity. To better understand the relative contributions of the different types of omics that were used in the validated model, they looked into the feature composition of this model. So there were 682 features that had non-zero contributions into the model, and they're shown here ranked based on their importance score. The top features do include all of the classes of analytes that were tested, and there were at least 2 features from each of the analytes that were found in the top 20. So this underscores the complementary information coming from the different types of omics and the importance of a multi-omic approach to enhance classifier performance. So in summary, Prognomic has conducted a large-scale plasma multi-omic study using a multi-site lung cancer case control cohort of 2,513 subjects for a total of over 800,000 orthogonal protein, transcript, and metabolite features that were tested and then used to train a machine learning model for a lung cancer classifier. This multi-omics classifier then validated in an independent holdout set, detecting both all-stage as well as early-stage lung cancer with high specificity and sensitivity. So I think this study highlights the opportunity to improve the sensitivity of blood tests for lung cancer early detection by incorporating multiple modes of data and machine learning tools. So these two examples indicate the potential that multimodal data and machine learning approaches can have to improve cancer detection in the lowest tumor burden settings, such as early detection and your disease immediately after surgery. There's types of approaches. There's also a lot of so I'd like to close by reflecting on some key areas for us as a field we explore beyond liquid biopsy, beyond ctDNA. The first of these is pre-analytics. Thanks to the efforts of groups like BLOODPAC, pre-analytic standards are now well established for plasma ctDNA. However, pre-analytic development for liquid biopsy analytes is much earlier for other liquid biopsy analytes. So additionally, it's well known that analytes like RNA or proteins are more labile than DNA. For example, it's generally well accepted in the diagnostic space that cell-free DNA can be stable at ambient conditions up to like 14 days. But proteins are pretty different. In this study of protein stability in K2-EDTA blood, the authors are reporting significant changes in protein integrity just hours after being stored at ambient conditions. And then even at -20, significant changes were seen in protein integrity on the time scale of days. So as we move forward with multi-omic tests, it will be critical for diagnostic developers that we establish guidances for best practices like BLOODPAC has already done for ctDNA. Analytics also pose a challenge here. Most liquid biopsy tests today rely on tumor-informed DNA mutation-specific assays, enabling high specificity, and giving us a way to sanity check the biological relevance of what we're detecting in blood. But as we move to other classes of analytes, they may not be unique to tumor. So for example, proteins that are highly upregulated in the cancer setting may also be upregulated in other physiological settings, like infection or even smoking. Therefore, it will be essential to thoroughly characterize the interpatient variation, both the demographic factors and environmental exposures, and to define appropriate reference ranges and establish new standards. And finally, multi-omics signatures and sophisticated AI approaches can be very powerful, as we've seen. However, complex models also come with the risk of overfitting and can fail to generalize beyond the training set. So to minimize these risks, our studies need to rigorously input only high-quality data and to use appropriate model and feature selection for the types and size of the data set that we're working with. We should insist on fully independent training and test sets, and then further validate models in prospective multi-site studies that include a diverse sampling of the clinically relevant patient population we're going to look at. Incorporating multi-omics and emerging analysis has tremendous potential to enhance liquid biopsy's test performance. These approaches may be able to push down the limit of detection that we have today with ctDNA, which would enable us to better detect cancers in low tumor burden settings, like early detection, right after surgery. So we need working groups like the one that we're discussing here to help us to establish best practices for these new types of assays, to help ensure reproducibility and to accelerate diagnostic development. Thank you.
Awesome. Thank you, Wendy. Much appreciated. And again, if you can take over the sharing screen right away. As we have seen with those two presentations, of course, with that multitude of parameters, analytes, and data that we all start integrating, we need to think about what are the toolkits so that not everybody reinvents this over and over. So Chris from Hopkins will actually tell us about a new analytics toolkit for building liquid biopsies. Chris, it's all yours.
Great, thanks. And just real quick, am I sharing my presenter mode or my actual?
Yes, you are in presenter mode.
So then I'll flip the.
No, you're perfect.
Okay, got it. All right, first, thanks BLOODPAC for having me. I'm really excited to be here, and it's an amazing attendance to see how many people are on the guest list. Today's talk is an analytical toolkit for building liquid biopsies. Before I begin, obviously, my disclosure slide. So a little bit about the talk today. I'd like to start with a brief survey of some of the most popular candidate biomarkers. And the structure of today's talk is a little bit of, what are the different types of statistical and analytical tools we can begin to use to get starting to evaluate candidate biomarkers. And once we started to work with them, how can we properly evaluate which of these candidate biomarkers warrant additional investigation? Next, I'd like to move into different clinical contexts and how this informs our selection of candidate biomarkers and how that will ultimately play in optimizing our algorithms. And I think most of the people are here is we'd like to be able to develop a diagnostic test that actually reaches patients and impacts human health. And so how can we go about structuring our studies to minimize overfitting and give our actual test the greatest chance for success? On this slide, you can see some of the most popular tumor-derived candidate biomarkers. I definitely missed plenty of them, but I just want to highlight these are the three biomarkers that our group typically focuses on. We typically look at genetic biomarkers, and we started to move into the fragmentation space. So if we look first at DNA mutations, you often see this analogy of DNA mutations are like searching for a needle in a haystack. When we sample a specific biofluid, we know that of the DNA molecules, most of those are derived from normal or healthy cells and will not have the somatic mutation of interest. And oftentimes the tumor is only shedding 1 in 1,000, 1 in 10,000, or even 1 in 100,000 mutant molecules. And so it often needs some type of toolkit to be able to find these rare events. And so you often see stochastic math and stochastics being thrown around in this space. And so by this, we just mean random sampling. So if we have a mutant molecule present at 1 in 10,000 DNA copies, how do we know if we pick the right 10,000, if we sample the appropriate 10,000 molecules? And so how do we build in some randomness into our analysis? Moving beyond just simple detection present or absent, we can layer a little bit more sophistication by doing some probabilistic modeling of the different mutation types. We know that if it's a transition versus transversion, this is going to have a slightly different probability. And we also know that genomic context can play a key role in the probability of observing a particular mutation. And also, ultimately, one of the key aspects of rare mutation detection is layering some sort of error modeling, whether it's PCR artifacts, sequencing artifacts, or library construction. We know that the errors we observe are not completely random, and we can start to model some of these particular variables to layer a little bit of sophistication about how we observe these types of rare mutations. Mutations have a series of advantages. These are a very well understood genetic event, and we can have a really good grasp of the events we're observing. We also know there are tied actionable therapeutics to the presence or absence of certain mutations, but these do come with a series of drawbacks, including, we're at an AI computational symposium, but these often require more complex chemistry and require labeling using molecular barcodes, often on both strands, and can face a series of challenges when we try to automate these protocols. These also require a large quantity of DNA. If we're searching for 1 in 10,000 molecules, that often means we need much greater than 10,000 molecules to analyze, necessitating a large quantity of blood to begin with. We also look at DNA copy number changes. This is often referred to as aneuploidy. On this slide, we can visualize what aneuploidy typically looks like in this setting. So we can visualize 1 through 22 of the different chromosomes. And on the on the y-axis, we can see some of the chromosomes either being gained or lost, as well as some of the smaller focal or subchromosomal changes that are occurring. This requires a very different skill set than looking at mutations, because oftentimes we rely heavily on some of the statistical tools needed to normalize GC bias. We know as the representation of the molecules throughout the genome amplify non-uniformly and require some type of GC correction. We also need to normalize across batches and control for some of the differences observed in large cohorts. But arguably the most important aspect is doing some type of change point analysis, whether it's circular binary segmentation algorithm or some type of hidden Markov model to identify when portions of the chromosome are either gained or lost. We know that not all the events we observed are these large chromosome or chromosome arm level events. Some of the key advantages for this type of biomarker is that it's a fairly well understood biomarker, and almost all cancer types, with depending on which review you look at, more than 90% of all tumors will have some degree of aneuploidy. So this means it can generalize to a lot of different cancer types. Because we're looking at a broad genetic event across the entire genome, we can reduce the quantity of DNA needed for this type of analyte. But it does come with some drawbacks, including the infinite number of possible breakpoints. So this means a lot of searching for different change points, which can add a layer of complexity and different number of possibilities that we're observing. We also have a challenge of interpreting outliers. Because aneuploidy is such a radical change to the genome, oftentimes outliers can be difficult to distinguish between true aneuploidy. Also want to highlight fragmentation patterns. I think in the past year or two, you've seen an explosion of manuscripts that have been looking at this type of candidate biomarker. Oftentimes, fragmentation or fragmentomics is looking at the size of tumor DNA compared to normal, but this can also refer to the motifs present at the starter end of tumor molecules, including recurrently protected regions and chromatin accessibility and a whole host of other phenomenon in the cell-free DNA. These can be often require some type of complex supervised machine learning where we predict or infer a class label based off of these feature sets. These can be incredibly powerful and really drive performance because you're able to learn hidden features, doing some type of discovery as opposed to relying on well-characterized genetic annotations. These, however, come with the downside that we don't necessarily have a causal mechanism associated with tumorigenesis, and we don't necessarily understand how or why these features are popping up. And this can also unintentionally cause our analytic methods to learn batch effects if we don't properly control for that, and unintentionally inflating our performance numbers and overfitting to a unique cohort. So if we zoom back, all of these biomarkers have large numbers of peer-reviewed manuscripts and all seem very promising. So how can we as an investigator and a practitioner of different AI methods go about which of these warrants additional investigation? So I kind of wanted to give a little toy example of how our group typically approaches how we evaluate which ones seem the most promising. On this slide, we have two different candidate biomarkers. We call them variable A and variable B. Red are the cases and blue are the controls. And we see a linear shift for variable A, but we see a bimodal shift for variable B. Both of these look pretty good, but is there a way to pick which one seems perhaps the most promising? I think most papers and most people will first start by generating an ROC curve and saying this one seems probably best. We also know that the computer scientists love to use mutual information, and this is another commonly used approach. We also can use accuracy, and when we measure the area under the curve, we can see a large difference between variable A and variable B. But one thing that often doesn't come up is that an area under the curve often emphasizes all levels of specificity, even those that are not necessarily useful in the practical sense. So our group has started to look at move away from using area under the curve, but only using a sensitivity at a particular specificity. For this, we just call it S at 98, meaning what is our sensitivity at 98% specificity? Almost all of our real-world applications require a minimum of 98% specificity. And so this gives us a way to distinguish those candidate biomarkers that may have high AUCs, but not necessarily work at the high specificities needed in this context. So we said the brief overview of the different biomarkers. We talked about the tools needed to evaluate them and how we can see which ones work. We also need to layer in how this is actually going to be used when the patients start to get these diagnostic approaches. We're at BLOODPAC, so I think most people associate liquid biopsies with blood, but there are a whole host of other biospecimens, whether it's urine, pap fluid, sampling the esophagus or cerebral spinal fluid, but each of these has different trade-offs when we go about designing our molecular approaches. Our group focuses a lot on multi-cancer early detection. This is in blood and usually applied with average risk individuals. So when average risk individuals receive some type of diagnostic approach, we need to do a really good job avoiding false positives. So we focus at sensitivities at ultra high specificities. You'll often see the positive predictive value is emphasized over rather than simply using sensitivity or specificity. And again, I want to highlight that moving away from ROC curves may be useful in this multi-cancer early detection setting. We have a handful of papers, but not enough time to kind of go into those details if you want to look more into that. When we look at some of the other biofluids, while we still need high specificity, we can kind of tone down the ultra high 99% specificity so that we can enhance some of the sensitivity, because for the most part, some of these other fluids rely on some sort of precondition, whether it's hematuria in the urine or whether it's GERD in esophagus or suspected imaging for cerebral spinal fluid. But these other fluids offer us the ability to trade off sensitivity and specificity. We have a handful of other publications at our group for these other fluids. But we've kind of looked at how we can possibly come up with or how we can tune our algorithms based off of the clinical context. But we ultimately need to do some type of seminal study that goes from our initial feasibility in the biomarker setting into coming up with a way to evaluate our diagnostic approach. And so typically people will assemble these samples and do cross-validation to assemble their performance numbers. But I really wanted to highlight this great study out of Freenome in 2019, where they looked at how cross-validation can kind of impact the evaluation of a particular study. Cross-validation simply refers to just randomly partitioning various portions of your data set into training and testing, and repeating this process until every sample has been withheld so you can properly evaluate your performance. And this is a hope that you can ultimately gauge how well you'll do in the prospective cohort. They do a nice job showing that oftentimes your feature vectors are correlated, and you can unintentionally learn aspects of the institution or the batch when you train and test on random partitions. And they do a really nice job of saying how this overestimates performance and propose a strategy where you can stratify by specific variables like controlling for the institution and batch. And we really wanted to build upon their idea for a lot of our future studies. So we've kind of taken a slight modification and called it our block strategy, where we, instead of using cross-validation, we have specific blocks where we do our feature selection and model training. And then at a separate time point, we freeze this aspect and move into our second block where we can really determine our biomarker integration and determine thresholds. We made a big push to rely on what is our metric for positivity and pre-specifying or determining our thresholds before we move into our validation cohort. We then, as necessary, move into our validation cohort and pair that with some type of publicly available external data set to evaluate how well our approach is working. And then at the conclusion of our study, moving into a reproducibility cohort where we can do technical replicates and evaluate not just the reproducibility of the assay, but the reproducibility of our algorithm. And so the ultimate goal is not necessarily to come up with the highest possible number in our studies, but to generate reliable, stable, and reproducible estimates, as well as evaluating how good our thresholds work at our desired performance metrics, so that when we go into our prospective study, we have a lot of confidence. Just want to highlight, this is the study where we kind of implement this strategy in using a machine learning approach where we integrate several hundred thousands of variables. To wrap up, we know that our evaluating candidate biomarkers requires a broad statistical skill set, and we wanted to highlight some of the key strategies we can go about in evaluating them. We also know that identifying promising candidate biomarkers can vary significantly depending on both the test statistic as well as the clinical context. And ultimately, I kind of want to use this time to say how overfitting really plays a huge role in the liquid biopsy community, and use this time to propose a couple strategies for both the BLOODPAC community and the larger field of how we can kind of go about tackling this overfitting issue. And so in this last slide, just wanted to wrap up with some just general questions of how we can go about tackling this idea. So the first idea that I wanted to address was that I think almost all publications nowadays require some form of external data set and orthogonal validation. I think any type of complex method really gains a lot of rigor and confidence from the community when you see that it works in cohorts outside of our own laboratories. The next is a little bit more of a nuanced point, but I think one of the key parts of this is oftentimes we read studies where you develop a model, and then in a later time point, there's a follow-on study, but the original model actually isn't described. There's some form of retraining and tuning, which masks some of the reproducibility issues. So I would propose to the community that anytime a model is developed, future studies must benchmark their current work based off of what was previously described, so we can gauge how well these models work in future studies and what the fall off is when we actually freeze them and go into prospective studies. And lastly, what are some of the required data elements needed to benchmark algorithm performance? I think I'm starting to run out of time, but I just wanted to thank, we have a huge group at Hopkins, and this includes a large number of clinical collaborators, experimentalists, graduate students, residents, fellows, and postdocs. And thanks for taking the time to listen to the talk.
Awesome. Thank you, Chris. Much appreciated. Bob, if you don't mind, or Chris, if you unshare, and then Bob can share. So now Bob is going to tell us how he has used his toolkit effectively, and is going to walk us through a multimodal example. Bob is Vice President of Bioinformatics at Tempus AI. Bob, the floor is yours.
Thank you very much, Peter. And I want to just take a minute to thank BLOODPAC for inviting me to speak today. Just before I get going, can everyone see my screen?
Yes, we can.
Great. Okay. So as Peter said, I am the Vice President of Bioinformatics at Tempus AI, and I'm here to talk to you today about the challenges and opportunities in leveraging multimodal and multi-omics data streams in diagnostics development. So why are we here? We're here today because liquid biopsy-based testing is largely confined to a single point in time and a single analyte, that is ctDNA-based genomic profiling. So where do we want to go? We want to go beyond a single analyte. We want to look at multiple molecular data sources, and we want to go beyond the blood tube. We want to look at modalities that are outside of next-generation sequencing or other molecular techniques. We want to look at other data streams that we can leverage to make informed decisions about patient care. So on the analyte side, we want to look at how we can use ctDNA in new and informative ways. We also want to see how we can integrate other common analytes such as cfRNA or the plasma proteome. And of course, we want to figure out how we can operationalize these multi-analyte tests. How do we get all these together at once? On the going beyond the blood tube side, we have a whole variety of measurements that are taken during the course of a patient's care journey that we can potentially leverage to help them make the appropriate therapeutic decisions. So we have things like radiography, clinical progress notes, therapy information, whole slide imaging, external testing, retrospective, whether it's the same reference lab or different reference labs. How do we integrate all these data streams to improve patient care, especially in the context of liquid biopsy? Apologies, I'm going to briefly advertise for Tempus and talk about some of our solutions. But the reason I do this is because I want to point to the commitment Tempus has made to having a variety of platforms that can be used to support patient care across the continuum, and also generate data that can be used in a multimodal capacity. So of course, largely we'll focus on our treatment selection assays today, but I do want to point to our minimal residual disease and therapy and disease monitoring assays, our XM and XM Personal Next, which is in collaboration with Personalis. And then our treatment selection assays, XT, our tissue comprehensive genomic profiling assay, XR, our RNA sequencing assay, and then we have XE, our whole exome assay. Of course, on the liquid biopsy ctDNA side, we also have XF and XF+, both of those therapy selection assays. Finally, we have the XG assay, powered by Ambry Genetics, which is a hereditary cancer risk assay. So today, I want to focus more closely on our tissue therapy selection assays, because this is where we've tackled this problem. We've had success tackling this problem in significant ways. So one of the key goals at Tempus is answering the question of how do we make diagnostics smarter? How do we make them beyond a point in time? How do we make them beyond a single test? How do we take all this information to say something about the patient? And on the tissue testing side, we have two assays using different analytes that are run concurrently in most cases. So we have our XT assay, tumor normal matched 648 gene DNA sequencing panel. And then we have our XR assay, which is an exome capture RNA sequencing test. It detects gene rearrangements, altered splicing events, and it does a transcript abundance analysis, sort of your classic suite of RNA sequencing outputs. And what kind of data can we generate from this? We can see here that we're getting data in many cases from one source and in some cases from multiple sources. We have a variety of data streams on the molecular side, and then we have data streams on the clinical side when it comes to a request for a test from a provider. We get direct integration with EHRs for a variety of hospital systems where we can utilize this data at testing time. We can also, of course, use our classic genomic profiling data, somatic mutations, germline mutations, tumor mutational burden, and other composite biomarkers like microsatellite instability. We also, of course, get a transcript abundance analysis across the expressed genes. At the same time, we also have an IHC service, so we can get PD-L1, HER2, MMR, a variety of different stains. So we're going to take all this data, and we're going to try to integrate it and say something useful. So this is sort of what everyone wants to happen, right? We want to have a big multimodal real-world database, and it contains molecular features across a variety of different testing modalities. We have clinical features, retrospective back to diagnosis time, and we have outcomes. So we have a nice big matrix of variables that we can jam into a big AI and make data science happen. Sadly, it's just never that easy. So here are some of the key challenges we see with respect to leveraging a lot of this information at test time. At the forefront, we have to work with providers to acquire much of this information. In the case of biospecimens, this is, of course, an actual procedure often. In the context of tissue, biopsy needs to be taken, needs to go to pathology, needs to be sectioned, needs to be reviewed, and needs to be prepared for send-out. On the blood collection side, of course, we need to schedule time with a phlebotomist for the patient. We need to draw blood. We need to get that ready for shipment. On the imaging side, let's say we want to use radiography. We need to have a recent scan from a given patient, and we need to have that data stored in a way within the hospital system so that it can be transmitted to a reference laboratory. The same is true of EHR data, though at a much smaller scale, where we need to have those data programmatically available so that we can leverage it. And this is purely a function of data exposure in the correct context. And then, of course, we have the logistics. The providers have the data ready to go. Now we need to get it. In a lot of the cases, this simply involves a truck or a plane moving between physical sites on the Earth. However, that introduces a lot of complexity. So now we need to get our blood tube, we need to get our tissue, and we need to get it from a provider somewhere on Earth to a reference laboratory. We also need to move data. This can be relatively straightforward, or it can be extremely complex, depending on the size of the data, the complexity of the data, how we're going to land it, where we're going to materialize it, a number of different factors that require us exchanging data both between hospital systems, EHR, and our operational processes. Once we get all this information, then we have our own operational processes, which need to be performed to mine the utility from this information. We need to perform image analysis. We need someone to take a H&E slide, and we need them to load it on a slide imager. We need to do our molecular workflows. Of course, when we get tissue, we have to run through our accessioning. Tech has to scrape the tumor tissue off a slide. Library construction, hybridization capture, DNA sequencing. In the context of XT and XR, this is actually 3 separate molecular workflows. We need the XT tumor DNA assay. We have the XT normal, the blood tube DNA assay. And then we have the XR assay, which is taking the same tumor sample and reprocessing it via RNA sequencing. And then, of course, we have a bunch of semi-structured data and imaging files we're getting from a provider, whether these be DICOMs, their own slide images, or what have you. So now we have to featureize this data effectively and harmonize it such that it can be applied to a specific multimodal, multi-analyte model. So now we need single feature pre-processing once we've even materialized it within our own ecosystem. So let's say we can get all these things going. We featureize our data, we generate our sequencing data, we generate our whole slide images, and we featureize the slide images. Then we have to do quality control. So, of course, each of these steps to ensure the data are of appropriately high quality need to go through some sort of quality control process. If you have 5 different modes as input, each of them require quality control. And if any of those modes fail, if your algorithm is not robust to some degree of sparsity, you will no longer be able to deliver a result to a patient. So you need to either figure out how to have an extremely high acceptance rate across all of your quality control methods, because of course these are combinatorial in terms of their failure rates, or you need to figure out how to do more with fewer data input streams. Of course, once we get through all this, we get through provider activities, we get through logistics, we get through operations, we get to finally put everything in our model to deliver a result for a patient. And that's where we get a report, and then of course we're able to send that back to a patient. So all of this has to occur within a clinically meaningful time frame. And by that we mean usually in the 7 to 14 days, some say 21 days, depends, time frame. And that's an exceptionally challenging ask. So here's where we summarize sort of the core challenges we have. We have to physically move biospecimens. We have to develop automated data structuring processes. We have to, of course, do our molecular testing. And then we need to have models before our big models to perform feature extraction, normalization, and prepare these data to be ingested into an algorithm. So we have multi-input pipelines that get us to the point of our final result or a classifier or what have you. And then, of course, we have to orchestrate all this cross-data stream integration. This is essentially an old problem in diagnostics. The problem is sensor fusion is hard. Now, AI and AI-based methods, especially Gen AI, there's lots of excitement there. And it's essentially a new crack at the same old problem. How do we do sensor fusion well? And there's some ability for AI to do that. And we're hoping it'll solve that, but we run into the same old problem. So I want to talk about where it can work. We can make this work with some intelligent decisions around what we're including in our training data, what we're including in our modeling, and what we're actually including in our test. Here is an example from Kyle Beauchamp's group at Tempus, where they developed where they're doing a clinical validation of what we call our immune profiling score, or IPS. This is a multi-omic algorithm that combines DNA data from our XT test and RNA data from our XR test into a single composite biomarker predicting response to immune checkpoint inhibitors in first or second line treatment. On the left, we have the inclusion-exclusion criteria, both for the clinical study. And on the right, we have the Kaplan-Meier curves reflecting patients who were rated as IPS high by the model or IPS low by the model. And we can see a very clear separation. I strongly recommend you check out the poster in the link. There's a further study of this biomarker compared to classical indicators of response to immune checkpoint inhibitors, such as tumor mutational burden or PD-L1, where we believe this method might actually be superior. So it allows us to take a lot of data that are usually generated at the same time because of our concurrent XT and XR testing, combine that with retrospective clinical data, and generate a useful test result for patients. So we're really excited about that. We want to see these kinds of things continue to grow, and we need to make good decisions around how we build these algorithms out to deliver successful results within clinically meaningful timeframes. So in summary, how can we take these learnings and extend them to liquid biopsy testing? Of course, one of the big ones is we need to generate more concurrent multi-analyte testing data. Right now, a lot of what we have is just ctDNA. We need to generate more data of different analyte types so that we're allowed to begin collecting all this information and beginning to model it. Of course, for that, we have to work with our pre-analytical variables. We have to discuss how we can appropriately quality control this data and move it through our processes. We also need enhanced clinical data availability at the time of testing. This is a computational logistics problem. How do we make the data available to testing companies or AI testing companies, allow them to ingest that data and utilize it within our models? And of course, more data is what we need. We need platforms that can generate large amounts of data, correlate those data to outcomes, and then figure out how we can apply those outcomes to an actual diagnostic test. And that where it comes to the final point, less data. How do we take our big models trained multi-analyte, multimodal, what have you, these large models, and transfer their learnings onto a smaller set of analytes? How do we take our learnings and make them more tractably applicable to our tests being run in a clinically meaningful time frame? And that's, I think, one of the key ways we'll overcome a lot of these challenges with making high complexity algorithms available as diagnostic tests. And that's what I'll leave you with. Thank you.
Super. Thank you, Bob. Much appreciated. If you don't mind sharing, and that was actually a great transition over to Jim Goodsey to take the panel discussion forward. We lost about 10 minutes, but as Lauren said, we promise you to finish at 4 p.m. sharp. So Jim, it's all yours.
Am I sharing okay? Am I in full, move to full presentation mode?
You're all good.
All good.
Now we'll get you.
So I'll be your host for the balance of the afternoon. And as your host, I'd like to start by a quick round of introductions of our esteemed panel of subject matter experts. And because we are short on time, let's do name, title, and area of expertise. Starting with you, Bob.
Hi, I'm Bob Grossman. I'm a faculty member at the University of Chicago, and my area is translational data science, machine learning, and AI.
Rebecca.
Hi, BLOODPAC. I'm Rebecca Leary from the Biomedical Research Arm of Novartis, where I work in oncology translational research. Here, I focus on what we can measure in patients in our clinical trials to advance individual programs, and what we can learn more broadly from patients to yield translational insights to fuel drug discovery. Thanks.
Aman.
Hi, I'm Amin Mazloum. I am the Vice President of Bioinformatics and Data Innovation at Exact Sciences. My area of expertise is actually model development and software.
And Asaf.
Hi everyone, Asaf Zbiran, formerly the founder and CEO of C2I Genomics, a MRD company, currently founder and CEO of Prism AI, a company that provides a platform for multi-omic data creation, harmonization, and analytics for disease and drug modeling. Thank you for inviting me.
All right, so let's jump right into our first two questions. And I think they really are important because they set a baseline for, I think, understanding the rest of the day's discussion points. So first question, it's now 8+ years, post the original FDA AACR sponsored liquid biopsy symposia. So why now is there this urgent need for multi-omic forms and approaches to liquid biopsy testing? Which of the technologies do you think will have the largest impact on the next generation of liquid biopsy tests? Asaf, could you take that question?
Yeah, thank you. First of all, I think that Peter and the other speaker give a great presentation of multi-omics and why do we need them and how to use them. A bit about my 2 cents. So our ultimate goal is to understand the disease, predict the future evolutionary trajectory, and from that, develop and assign the right treatment to the right patients. We all understand that biology and especially cancer are complex. So cancer evolution is not done in a vacuum. The evolution of the disease is a network of dependencies between the cancer somatic landscape with immune system, with the host microenvironment, with metabolism, with other signaling. Multi-omics and multimodal measurement allow us a more holistic view of all of the different processes. And that allows us better understanding and better assignment or personalized medicine of cancer treatment. I think it's not necessarily the need, but the opportunity. If we look on beside the multi-omic methods, which are DNA, RNA methylation, proteomics, what we have seen in the last 10 years is a huge accessibility to different sources of signal. So now we can separate between the tumor biopsy, circulated tumor DNA, the immune cells, PBMC, ex vivo organoids, tissue digital pathology, blood-based or liquid digital pathologies. Each one of these modalities provide a very different snapshot of the behavior or the system that are underlying the disease. The big challenge that we have now is how to integrate, harmonize, and build multimodal models and biomarkers from all of these signals. And again, there is an opportunity. The risk is also the increased number of features, increase the risk of overfitting, batch effect. Some of the speakers discussed batch effect, and I think we are going to talk about it more later on.
Very good, Asaf. Thank you so much. Question number 2, and Rebecca, could you field this one? How do you view the role and added value of multi-omics in cancer screening and treatment management? And in what ways could AI complement liquid biopsy testing for more accurate cancer care?
It's a great question. And if we're thinking about cancer screening and treatment management, as Chris pointed out in his excellent talk, at the end of the day, what we really care about is sensitivity and specificity to detect any residual disease in treatment management and to avoid overdiagnosis in the screening setting. This can be addressed both by multimodal and AI approaches, either separately or in combination. By looking at new or differentiated sources of cancer signal, as Asaf just mentioned, we can augment what we're learning from conventional ctDNA assessments. When we're employing advanced computational methods, we can leverage the wealth of data generated in this field over the past close to decade. And BLOODPAC is the place that I look to in support of bringing these data together. In combination with multimodal data and AI, we can use AI to extract signal from multi-analyte data sets to enhance sensitivity and specificity, impacting both cancer screening and treatment management settings. Additionally, there are some emerging diagnostic modalities that are starting to shed light on new ways liquid biopsies can be used for precision medicine. So going beyond sensitivity and specificity for the presence or absence of a cancer signal, and specifically getting to inferred expression to look at expression of cell surface targets and non-genetic mechanisms of resistance. And I'm especially excited to see how kind of the nascent technology space of this new kind of component of multimodal analysis can be bolstered by advances in data analysis to move these approaches towards clinically relevant ctDNA fractions.
Thank you, Rebecca. So Bob Grossman, for the sake of many in our audience, could you please explain the differences between multimodal machine learning, multimodal AI, et cetera, et cetera?
Yeah, thank you. So first of all, the easy part of that is multimodal. Everyone knows multimodal. It's when you take different modalities of data expression data, proteomics data, clinical data, image data, H&E stained images, et cetera, and combine them. The harder thing to distinguish is the machine learning, deep learning, AI, because there's a real difference in terms of what are the techniques, and then there's a marketing difference in that techniques which just used to be machine learning are now AI. But on a more serious note, I think I would think of the difference between ML and AI in the following way. You have basic statistical models, which everyone uses and are very good at combining multimodal data. You have also machine learning models, which introduce a wider variety of techniques for combining multimodal data. They may be tree-based, support vector machines, other techniques that have been around quite a while and are very well understood and can be used to combine multimodal data. And that includes basic neural nets. They're not often used for various reasons, but they are also a core machine learning technique. Then you have deep learning, is when you take neural nets and you take many, many layers and you create models that have a lot of parameters, millions of parameters or more, billions of parameters. And they can be very powerful, especially with images and other types of data. But that was the deep learning that was introduced about a decade ago now. More recently, we've added techniques to deep learning. I think everyone's heard the terms attention and transformer, which makes deep learning very effective or more effective for sequence data, for imaging data, et cetera. These are the large language models, foundation models, et cetera, when you take deep learning and you enhance them with these techniques to create the current generation of models, which are based on large language models and / foundation models. And finally, for each of those generations, we've created techniques for combining multimodal data, including most recently with the large language models, which have been used quite successfully to look at images and clinical data, images, expression, et cetera, and are increasingly were referred to today. One warning, you can use more sophisticated techniques with more parameters moving from deep learning to foundation models, but if a signal only appears as you change the technique, in general, I think you should be suspicious. If it's a real signal, even simple models will pick it up. The advantage of going to these more complex models is that they can improve the characteristics of the model. So you have increased specificity, sensitivity. You might have asset 98 and those kinds of things that might be better. So that's just something to keep in mind as you think about the difference between multimodal ML, multimodal deep learning DL, and multimodal foundation models.
Bob, awesome job with that tough question. All right, Amin, what are your thoughts with regard to, where and at what stages can AI help, during product development of a liquid biopsy test?
Sure. So I want to just step back and mention that we were all looking into different modalities of data that we can bring more information, or by measuring different analytes or class of analytes, bring orthogonal information that can improve our accuracy in predicting the indication or seeing that earlier in time. Now, the real value of all of these analytes will materialize if we can provide them to the bedside, biomarkers to bedside at scale, with a reasonable cost. And we do know that the drug development is a labor-intensive, costly, and time-consuming effort to take those biomarkers from science with technology to the bedside. Where we can use AI to reduce this cost and time would be a big opportunity from clinical trial design, from inclusion and exclusion, using AI to identify the patients for enrollment, using AI to identify biomarkers, as many people here described that. And when it goes into development in automation for controlling the instrument, for identifying the errors in the instrument, and when you go to the operation you want to scale, identifying the modality of the operations, when and where things are going wrong before the impact has happened. So these are some of the areas that AI could be used to not only reduce the time of the development, the cost of the development, but also help them to scale. Another area that it can really help is in the software. So medical device that we build, it's again, building the software is time consuming, it's error prone, but building high quality software requires a lot of effort. Now the tools allows us to build more code in a short time with following better practices, building more test cases, so avoiding downstream bugs that is costly to repair and could be impactful to the resulting. Also in generation of an actionable results. It was pointed out that we identify a lot of mutations in the tumor profiling. Not all of them are relevant. Which one are actionable? Right. This is a labor-intensive activity, but really sometimes result in poor user experience because of the time that it takes and the inconsistency that it can bring. So using AI tools to delivering a more reliable, a more consistent, actionable mutation results to the providers, to the patients, would be an opportunity to again, bring the impact of those biomarkers to the bedside in a shorter, at scale, with a higher quality to the patients.
Thanks so much. So Rebecca, so as our pharmaceutical subject matter expert, how do you see, multimodal analytics and AI approaches in liquid biopsy testing advancing pharmaceutical development and supporting clinical trial design?
Yeah, over the past decade, we've seen the huge impact that just kind of conventional liquid biopsy analysis has made in the translational research biopharma space. And there are a bunch of places where AI touches outside of liquid biopsy around patient enrollment and clinical trial design that I won't touch on here because it's kind of outside the field of molecular diagnostics, but all super important applications of AI to speed up evaluation of experimental therapeutics and get them to patients faster. When we're thinking around liquid biopsy and multimodal analysis overlaid with AI in the clinical trial setting, there are three areas that I am really focused on. I'm going to say it again, improving assay sensitivity, especially for tumor agnostic or liquid-only assays. And here there are some very real logistical challenges, as Bob Tell mentioned in his talk, in terms of just even getting the tumor tissue. So we need to improve assay sensitivity in this setting to hopefully support dose optimization efforts and to provide real-time data to guide decision-making in first-in-human clinical trials, especially when there are multiple potential indications being evaluated or multiple combination partners that we have an option to expand it. The second is enhancing the role of liquid biopsy as a surrogate for tissue biopsy. Just said that getting tissue is really hard. And when we get tissue, it's almost always only archival or a baseline, very occasionally a single on-treatment biopsy. And we're desperately interested in learning about how our therapy is impacting the tumor and the tumor microenvironment. So anything that we can get from liquid biopsy in order to understand pharmacodynamic markers and the impact that our therapies are having in the tumor and TME is really important. We already know that liquid biopsy and tissue mutation profiling are highly correlated, but we want to extend what we can learn from the periphery to include cell surface target expression and how our therapies are modulating the TME. And then the third is increasing the speed by which we're able to evaluate our novel therapies in the settings where they're likely to have the greatest clinical benefit. And here I mean in the neoadjuvant or adjuvant setting for earlier stages of disease. These are generally really, really big trials that take a very long time to run. And as liquid biopsy matures and the sensitivity and specificity are further improved by incorporating signal from different analytes and by advanced computational tools, these advances in liquid biopsy can be brought into the biopharma space to help us enroll patients for smaller trials in these settings, and as MRD as a surrogate endpoint to speed development times, not only to make drug development more efficient, but to get these exciting new therapies to patients faster. Thanks.
Thank you, Rebecca. So moving on, and so Bob, I wanted Bob Tell bring you back in from the Speaker Bureau to kind of take this next question, because I thought I kind of read on top of, the recent launch by Tempus of your beta version of Olivia, which is a AI-enabled personal health concierge app. So tell us how AI can help with interactions with patients, the associated clinical outcome data. Give us some thoughts on that.
Thanks, Jim. Great question. From our perspective, there are a lot of opportunities where we can help both in terms of clinical trials and in terms of how patients interact with their own healthcare data. One of the challenges we see a lot with patients is simply the same problem we see with oncologists: the sheer volume of data they're getting thrown at them. They want to get a second opinion. Sometimes they have to get physical printed paper generated to bring to another physician. They sometimes have to get data from multiple sources. They need DICOM images. They need NGS-based sequencing reports, other diagnostic methods, pathology reports. These are all over the place and can be difficult to collate, to utilize, to understand. This is where a lot of these tools can be exceptionally valuable, because, of course, these AI-based tools are excellent at interpreting a human-generated request, finding the things the person wants, and then potentially adding a layer of interpretation on top. So we see a lot of value with both helping patients collate their data, helping patients understand their data, and helping patients work through their care journey, both in terms of questions like, I have an appointment with my oncologist in 2 days. What question should I ask? As we begin to develop high-dimensional representations of a given patient, we can potentially inform them best as to what questions they need to ask, both based on standard of care recommendations and based on their personal molecular and multimodal profile. So there's lots of opportunities there, both for the patient and potentially for people looking to develop clinical trials with using these high-dimensional representations to optimize next steps, optimize selection of patients, and ensuring everyone has all of the information available to them that they need to make the right decision at any given time.
Thanks, Bob. That's great. So moving on, Amin, so what are your thoughts about how we can utilize multi-omics and liquid biopsy testing to enhance prediction care by integrating clinical data with primary tissue samples?
Sure. So twofold, I'll try to answer this in twofold. Number one, being able to access the clinical data, which basically includes the history of the patient's records. That is a challenge, because as we know that the more data we ask for, the more sparse the data will be. So one solution is to how we can better verify data. The standardization of the clinical data is in my opinion, is a kind of a wishful thinking, but how we can make the data more, we can more democratize the data. And that starts thinking of us being becoming a donor of data in our community. So to me, moving away from holding onto my data because it is mine and I could be manipulated if my data is get access to the wrong hands. It has elements of truth. But if we really want to benefit from the data and have the community benefit from our data, we have to be moving the mindset toward democratization of the data. Of course, with all sort of privacy and security is necessary. But really making the data of every single patient becoming available for research, that's basic data availability, and also being able to connect the sources of data in a way that we can integrate the clinical data with analytical data. Because we do know when you move to the multi-omics data, there are more elements in the patient history and life that can impact those data that if we don't have access to, it becomes challenging. Because a very simple example of that is the methylation or methylome, which could be impacted by many factors, including the lifestyle or the history of for example, smoking, right? So in order to be able to benefit from the clinical data, we need to be able to access those, learn from those, and train the models with those. But on the model side, as I think Bob pointed out, we have to make our models such that they are more prone to more sparsity. Because the more dimensional the data, there's a higher chance of sparsity of the data. So being able to verify the data, democratize the data so people can learn and outlive other people can outlive from my data when I'm still alive. I mean, I just want to compare that to organ donors, right? So many of us just put that organ donor in our driver's license, yeah. So if we were not able to leave for whatever reason, somebody else could leave beyond us by using our organs. I would say we should use the same concept for data. Why the community cannot benefit from our data by providing us those who can learn from them. So a kind of a mindset shift which is not simple, but that needs to happen, especially now in the world of AI that requires a lot of data to be able to train our models. Number two, being able to integrate those in the model, but also making models, as was pointed out, that are robust to data sparsity. And again, this is not necessarily related to the tissue data. I'm talking about any analyze that we measure and all the elements of clinical data that we can get access to that can influence the decision-making post-analytical measurements.
Thanks, Amin. And we're running short on time, so I'm going to jump to Asaf with the question of what are your thoughts as to the potential to engage multi-omics in managing clinical indications beyond cancer, such as autoimmune, neurodegenerative, and inflammatory disease? You're on mute, Asaf.
Yeah, sorry. So any tissue damage is secreted in the blood. So we can use cell-free DNA to diagnose a large variety of diseases. Also, if we are looking at autoimmunity, mental health, inflammatory disease, neurodegenerative, all of these diseases have a immune signature that we can find in the PBMC. The signal for all of these is not a DNA mutation. It's more the transcriptome and methylation. So definitely using a multi-omic transcriptome, methylome in the cell-free DNA in the PBMC is going to open a large variety of opportunity for screening of different diseases. I will also mention another point regarding cancer treatment management. We have seen an amazing adoption of MRD for cancer treatment management. We saw recently the endorsement of or acknowledgement of the FDA for MRD as a potential surrogate marker. We believe that with multi-omic, we can actually extend it to prediction of the evolutionary trajectory of the disease. So imagine that besides just saying what the probability of recurrence for early stage patient, you can actually predict what is the site of recurrence, which organ is going to have metastatic site. If you can predict that, then you can follow up with organ-specific surveillance for curative surgery, organ-specific therapeutic intervention. So this is one example how multi-omic can basically going to open a large new spectrum of opportunity for treatment management.
Thanks, Asaf. So running short on time. So what I thought we could do with the next 10 minutes is spend 5 minutes taking questions from the chat that our audience had provided. And, our first question comes from the FDA from Anan that came during Wendy's presentation. And the question was, what was the specificity for the multi-omics-based screening? Wendy, could you take that?
Sure. So in the data slide that I showed, the specificity for the training set was 91%, and in the test set, it was 89%.
And then we had a question from Daria for Chris. And basically, she's saying, Chris, you're preaching to the choir, but the impact of technical artifacts and the training validation cohort settings with variable pre-analytical factors has to be fully understood and a complex biomarker signature development before any interpretation of the performance data can be made from a clinical perspective. Just reflecting on the prognomic analysis, has any of that assessment been done informative in the signature development? So maybe that's a Chris and Wendy response.
Yeah, definitely. I think it's a great point, and I've seen this happen in a lot of couple larger manuscripts where people will come out with a model and then try to benchmark it on an external data set, and it obviously has a lot of difficulty. What we had found is, there's different ways of demonstrating reproducibility in an external cohort. There's the model, which is everything's frozen, and that makes a lot of sense, and that's kind of a pipe dream that often doesn't necessarily work given all the external pre-analytic conditions. But if you can distill down even just trends, so what we did in our manuscript was it was a completely orthogonal experimental technique, but we showed that some of, so the paper was on allosigns, you can show that some are either elevated or increased or decreased, and that was at least sufficient enough to show that the trends replicated in other people's hands. But you're absolutely right, the models oftentimes are too challenging across experimental techniques. But if there's one way to distill it down to a couple observations or variables.
We got a question from Don for Bob Grossman. Please comment on LLM scaling and the emergent abilities, unexpected capabilities, which are currently not understood, and how these may help with multimodal data understanding. So Don and Bob both, we need to define LLM scaling first.
Which Bob, is that Bob Grossman or Bob?
Bob Grossman, yes.
Okay.
Yeah.
So LLM scaling, I mean, if normally scaling is used for large language models as this more or less remarkable observation that as the number of parameters in an LLM grows from absurd to very absurd, from billions to 10 billion to tens of billions to hundreds of billions or a trillion parameters, the model improves on standard benchmarks. This scaling law, which leads to all the current frontier models needing more and more data, is the reason that they get better, but it's also the reason that the largest frontier models are running out of data. So, and having to move to synthetic data and scraping data and cross-licensing data. So I would phrase this slightly differently, especially for this community, in which I would expect, and we're doing research on this and others. First of all, there are multiple scaling laws. There's the one I just described. There's also the scaling law when you fix compute, what's the right amount of data? I think for this community and something I'm working on and others are, is a scaling law when you have high quality data, because a lot of the data for the large language models and foundation models are low quality. And unfortunately, the quality is getting lower as ad tech is used by AI to generate more and more low quality data. But in our community, when you have high quality curated data, you can expect to have smaller, so-called small large language models or mid-scale large language models that can compensate the number of parameters by for the quality of data. So that's another scaling law. And I expect we'll see lots of other scaling laws because this is a brand new set of observations with the theory lagging behind. I'm conscious of time, so I'll stop there.
Thank you, Bob. So I'm going to close our portion, and I have two comments. The first is, I can't thank Peter enough for his great introductory comments. Our Bureau of Expert Speakers, our panel of subject matter experts, for taking time away from their work. I know you're all very busy, to share your vast amounts of knowledge with the audience today. So really just can't thank you enough. Awesome presentations. And then finally, our ask. As we close out the seminar, I have an ask for the audience is to use the chat or, follow up later in person or by email, with your thoughts, suggestions for topics that you feel the BLOODPAC could actively pursue in the form of a working group. Working groups are how we contribute to the, scientific community. Focused on one or more of the above topics that were discussed today that could make a significant positive industry-wide impact on accelerating the adoption of multi-omic AI-driven liquid biopsy testing into clinical practice. So, please take a moment to use the chat, send us an email, see us in person. Love to have your feedback to take us to the next we'll say phase of our BLOODPAC programs. So I'm going to turn it over to Peter and Lauren for closing out the session.
Great, thank you, Jim. Much appreciated. And just to echo what Jim was just saying, and Wendy mentioned this briefly, the goal here is really to take this, and I'm sure that Lauren will echo this, take this discussion, because this is a topic that has to be front and center for all of us, and figure out as BLOODPAC, how do we contribute? How does this work? And with that, I hand it over to Lauren, because I think this is a really exciting opportunity for us as a community to do something together. Lauren?
No, I'm thrilled. Thank you all for participating. The lists of questions were terrific. Happy to have anyone shout stuff out now. I feel like people tend to do that on our calls towards the end. But just, again, echoing what Jim and Peter said and what we've really been aiming towards just as quick background one more time. These seminars are wonderful, but they also have a purpose, and that purpose is to decide and help our scientific co-chair committee make a decision about whether or not to invite individuals to submit a proposal to actually form a working group. So any of your input, as Jim and Peter just mentioned, is incredibly valuable to us. And the more that I learn about this topic, the more I learn there are a lot of different areas that we could, and directions we could go in forming a working group that isn't just a think tank, but is a group that provides a deliverable to the community. So it's necessary to actually hone in on exactly what it is we are thinking about working on and what we would deliver to the community as part of that product. I just want to remind everyone again that this seminar will be available in its entirety on our website shortly after we finish. Not today, but in the next week or two. We will also be coming out with a white paper recapping all of the information. Any questions that you felt you didn't get answered, but you put in the chat, we will be answering them, asking our panelists and speakers to participate in answering them in a written format for that white paper. And if you have any questions that you would like us to address that you didn't get a chance to type in, send them to me. I'm happy to get them answered and include them in the white paper as well. With that, I promise to get everyone out of here by 4 o'clock. I want a tremendous thank you to Sam, Wendy, Chris, and Bob Tell for being speakers today. You have gone above and beyond. I want everyone to know that the work that went into this was tremendous. This isn't just please put together a presentation and show up. They spent months, literally months, sitting together, discussing what would be the hot topics of the moment and what they should focus on and make sure no one was overlapping, and really thinking through what opportunities there were in the field. So thank you for your commitment to doing that. To our panelists, Rebecca, Bob Grossman, Amin, and Asaf, you did the same thing. You sat through those discussions. It was incredibly valuable, and we can't wait to hopefully work with you in the future on this topic. A huge thanks to our BLOODPAC co-chairs, moderators, and co-hosts, everything for this entire seminar. Jim and Peter, you two also, thank you for thinking about what this could look like and bringing it to fruition. And finally, a round of applause, if possible, even in silence, for Doris Lopez, who did coordinate this entire event, who this would not have happened without her. And Cheyenne, who's actually making it all look beautiful in the backgrounds. So thank you, everyone. You have 2 minutes back of your day. Please feel free, call, email, text. We're here. We want to hear from you. Thank you so much. Have a great rest of your afternoon.
Thank you, Lauren. Thank you, everybody. That was awesome.
Bravo.
Thank you. Thank you.
Thank you.